In silico analysis of various antiviral compounds against spike protein of COVID-19 using docking methods. Abstract: the current examination assessed the adequacy of hostile to viral compounds against the objective protein, spike protein of COVID-19 utilizing in silico approach. Structure-based docking studies was performed using PyRx 8.0 tool to retrieve potential candidates with high affinities binding against antiviral compound models with several targeted proteins repositories. The chosen target protein was 6Lu7. Molecular docking was carried out using various antiviral compounds with the target protein of microorganisms.Free. Vitality of restricting was recorded for the compound with its objective. 9 different compounds were docked with the target protein, within which was found to own higher binding affinity. Therefore, the current study plays a guiding role in developing new inhibitors with better binding affinities with the virulent protein of pathogens, followed by invention of novel drug to treat viral infections. Keywords: spike protein, COVID-19, PyRx 8.0. 1. Introduction 1.1 S protein. Pneumonia of unknown cause was detected in Wuhan of Hubei province in China and reported first on December 31, 2019. 3. On 30 January 2020 the outbreak was declared as public health emergency of international concern. On 11 February 2020 the new coronavirus disease was named as COVID-19 by World Health Organization, WHO. The WHO declared it as world pandemic on March 11, 2020. In exception to Antarctic the virus contact the majority parts of the globe. 9. COVID-19 is a communicable disease caused by newly discovered coronavirus. These are RNA group virus which will infect mammals and birds. 6. This caused the intense tract infection. There's no medication or vaccine yet discovered to forestall the human corona infectious. The National Institute of Health, NIH, stated that this virus is believed to own genetic similarity to Bet coronavirus 8. The virus either directly via close contact and respiratory droplets of sneeze and cough or indirectly via contact through contaminated surfaces. This can be associated with SARS, severe acute respiratory syndrome, and MERS, Middle East respiratory syndrome. The symptoms of coronavirus include fever, dry cough, and difficulty in breathing. 9. It takes a minimum of 5 to 6 days to indicate symptoms when the person is affected from virus. The foremost promising test for the identification of virus is RT-PCR technique. The COVID-19 belongs to the subfamily Orthocoronavirini within the family Coronaviridae, order Nidovirales, and realm Riboviria. COVID-19 is an enveloped, positive-sense single-stranded RNA genome encoding quite 20 proteins. This has higher binding affinity with human ACE2. This is often wrapped during icosahedral protein shell. The genome size of COVID ranges from around 26 to 32 kilobases, one among the greatest among RNA viruses. They need characteristic club-shaped spikes that project from their surface, which in electron micrographs create a picture paying homage to the solar corona, from which their name derives. 12. Novel approach of drug design and docking studies may be utilized for the invention of some therapeutic drug candidates against COVID-19. These in silico techniques can minimize the time and efforts within the pipeline of drug discovery. These techniques include study of the protein structure answerable for the disease or disorder, then identifying a ligand molecule which may have potent activities. 1. Molecular docking techniques are study of interaction of the ligand with the molecule at situation. The site is that three-dimensional site of molecule which directly affects the activity of the protein. Although the activity is often either positive or negative, but still it helps medicinal chemists to proceed on rational path. 7. The interaction ligands are measured as binding affinity at receptor binding sites. The less the binding affinity to the receptor at the prescribed site is, that the more practical. For measuring certain values, the charm force fields are used. Although the values differ marginally from system to system, and from software to software. This is often due to the variations within the system's genetic algorithm. 7. We performed the docking of some antiviral compounds with COVID-19 spike protein main protease in complex with an inhibitor N3. The S protein, spike protein, consists of an ectodomain region, ED, intracellular domain, and trademark region. It's a kind I transmembrane, trademark, protein which appears as clove-shaped. The ED region consists of 2 receptor binding domains, RBD, S1 and trimeric stalk containing S2 subunit associated on C-terminal 4, 5. When S protein associates, the virion appears as trimeric and appears sort of a crown structure. This structure is termed as coronavirus. The S protein is found to own potential role in viral entry inside the host. The activation of host immune repose against the virus by S protein has been reported. This protein is taken into account as a possible target for drug discovery because S1 domain and host ACE2 for SARS-CoV and dipeptidyl peptidase 4, DPP4, form ERS-CoV related to host and viral membrane fusion mediated by S2 segment potentiate the CoV to release its RNA in host cell 10. 2 Materials and methods. 2.1 Preparation of receptor. 2.2 Preparation of ligand. 2.3 ADME studies. 2.4 PyRx. The target protein, spike protein of COVID-19 was obtained from PBD database, www.rcsb.org. The SDF file format of PBD ID, 6LU7 was downloaded for docking studies. PubChem compound database contains approved synthetic portrayal data gave to clarify substances in PubChem substance. Structures stored within PubChem compounds are pre-clustered and cross-referenced by identity and similarity groups. Theaflavin, CID, 135403798, theaflavin 3 gallate, TF2A, CID, 7Q307578, theaflavin 3-3 digallate, CID, 135403795, hydroxychloroquine, CID, 3652, zanamivir, CID, 60855, peramivir, CID, 154234, baloxavir marboxil, CID, 12408186, oseltamivir phosphate, CID, 78000, galanin, CID, 5281616. Structures were retrieved from the PubChem compound database. Swiss ADME studies are used for predicting the pharmacokinetics and physicochemical properties. Molecules that follow the Lipinski's rule with no violations are listed. The method involves the assessment of absorption, distribution, metabolism, and excretion, ADME. PyRx could be a virtual screening programming for computational medication revelation which will be utilized to screen libraries of mixes against potential medication targets. The macromolecules are docked with small molecules with the assistance of virtual screening method. The PC-aided docking method is extremely useful in identifying the lead drug compound and made the drug discovery procedure easier. PyRx may be a Python artificial language which will run in any modern computer from PC to supercomputer too. 3 Result, 3.1 Target and ligand preparation. 3.2 Virtual screening. 3.3 ADME studies. The Swiss ADME studies were performed for the library of ligand molecules. The ADME properties of H donor, H acceptor, log p-value, and molecular weight were determined. Some compounds violated these drug likeness properties. The non-violated compounds are then used for future studies. Compound. For discussion. The virtual screening and ADME studies reveal that galanin has higher binding of -6.8 kcal/mol as compared with other compounds. Although theaflavin 3 gallate has higher binding affinity of -8.5 kcal/mol, it doesn't satisfy the drug likeness properties. Other compounds like hydroxychloroquine has binding affinity of -4.9 kcal/mol, zanamivir has binding affinity of -5.5 kcal/mol, peramivir has binding affinity of -5.5 kcal/mol and oseltamivir phosphate has binding affinity of -4 kcal/mol. 5 Conclusion. The in silico screening of biologically active compounds were distributed towards the spike protein of COVID-19. The molecular docking studies revealed that galanin showed higher binding affinity by satisfying ADME properties. The interaction study helped us to grasp that galanin may be employed in drug discovery process. Further in vivo studies are often applied to search out the character of the ligand molecule in future.